Hi everyone, my name is Ikta, and today we are diving into major technological breakthrough in genomes, a new method called SDR sequence or single cell DNA RNA sequencing. This tackles most persistent challenge in genomes, understanding the function of the vast non-coding region of our DNA, the dark matter of the genome. The core problem is linking genetic variations in non-coding regions directly to their functional consequences on gene expression, and single-cell DNA RNA sequencing is the tool that finally allows us to do this at scale. For over a decade, GWAS confirmed that most genetic variations associated with complex disease like cancer disease, heart disease, are not in the genes that code for proteins. In fact, more than 95% of disease associated variants, the variants we need to understand, are non coding regions. These are the regulatory elements that act as volume and on-off switches for our genes. A subtle change, like single SNP in an enhancer, can alter how a transcription factor binds, subtly modulating a distant gene's expression. When compounded, this is the root of complex polygenic diseases. The complexity of disease means we need single cell resolution. Why? Because a variance effect is often highly specific. If you sequence a whole tissue sample, which is a mix of millions of cells, you get an average signal. That average completely masks a variance effect that might only be active in small yet critical subpopulation of cells. To solve complex disease etiology, we need to bridge the gap. We must link the specific DNA change to its exact functional output inside one single cell. Um, Before SDR sequencing, single cell multiomics was limited in two crucial ways. First, low through output, analyzing only hundreds of cells isn't only enough to capture heterogeneity of a tumor or brain sample. More critically, there was a systematic non coding bias. Older technologies were bad at capturing variants in non coding regions. They were designed to find variants that were already expressed as RNA, essentially forcing researchers to ignore the 95% of disease linked DNA that is non coding. Researchers were limited to studying the small fraction of coding variants, while the critical regulatory code remained locked and inaccessible. Um, Single cell DNA RNA sequencing developed by Linderhofer and colleagues is a specialized multi-omic sequences pipeline that provides necessary leap in scale and sensitivity. The key technology called breakthrough uses droplet system, nanoliter oil water chambers, to encapsulate single cell within that tiny droplet. The DNA and RNA are processed at the same time. The critical step, dual barcoding, by applying the exact same unique molecular identifier to the DNA and RNA of that cell. The researchers can computationally link the specific non-coding genomic variant to its resulting expression profile. This not only links the data but also provides high throughput, non-biased capture necessary for studying complex tissues. This is the figure of endocytosis. The initial validation of SDR sequencing was performed using beta cell lymphoma uh, patient samples, which are ideal because they harbor significant varied sets of genetic mutations. Using SDR sequencing, the researchers performed functional phenotyping. They saw the direct functional outcome of the variant. The critical finding was a clear mechanistic link. Beta cell carrying an increasing burden of genetic variants, particularly those likely in non-coding regulatory regions, exhibited stronger activation and proliferation signals. In simple terms, genetic accumulation drives the cancer towards a more aggressive phenotype. This is a monumental step. It's not just an association, it's a demonstration of mechanical at a single cell level, allowing us to interpret the entire 98% of the regulatory genome. In conclusion, the introduction of SDR sequencing is a game changer. It has solved the long-standing technological bottleneck by providing a high-throughput, non-biased method to link non-coding DNA variants directly to their functional output in thousands of single cells. This technology will fundamentally accelerate our mechanistic understanding of polygenic disorders and usher in a new era of highly precise diagnostics. Uh, we are transitioning from interpreting the small 2% of protein coding genomes to finally mapping and understanding the regulatory logic of their entire 98%. Thank you.